Welcome to the second lecture associated with muscle tissue. Today we're going to be going over microscopic anatomy of the skeletal muscle fiber. Remember, we're going to be using the skeletal muscle fiber as our model for the construction of muscle tissue in general, although when we talk about smooth muscle and cardiac muscle, we will be comparing it back to the skeletal muscle fiber. So we call a skeletal muscle fiber a fiber because it's very long, cylindrical, and it looks somewhat like a rope, especially when we see the interior structures of the individual cell. The term skeletal muscle fiber is a synonym. It means the same thing as a skeletal muscle cell. The skeletal muscle fiber is made from the fusion of many stem cells. All of these stem cells have individual nuclei, so the whole cells fuse together and the nuclei are pushed up to the edge of the cell, so there are multiple peripheral nuclei. There's a special set of terms that we often use in association with muscle tissue. The term sarco is a prefix specifically referring to the muscle cell. Plasma membrane has a synonym. It's called the plasma lemma. So the term lemma, L-E-M-M-A, means the same thing as membrane. So specifically, the sarcolemma is the plasma membrane of the skeletal muscle cell. Even more specifically, it's the cell membrane material that covers the outermost area of the skeletal muscle fiber. We will later on be looking at an extension of the cell membrane that goes internal, that goes into the cell itself, and that's going to be known as the T-tubule or transverse tubule. Another special term that we use in association with the skeletal muscle fiber is the term sarcoplasm, which means the same thing as the cytoplasm. There are some specialized organelles for the skeletal muscle fiber, one of which is the glycosome. The glycosome is used for glycogen storage. Glycogen is a carbohydrate. It is the polysaccharide made out of glucose. So it is how the humans will store glucose. Glucose is the energy molecule used best by the cell. Another structure that will be found in the sarcoplasm, found specifically in muscle tissue, is myoglobin. Myoglobin is very similar to another molecule found in red blood cells known as hemoglobin. Hemoglobin and myoglobin both store oxygen. So this is another indication that the skeletal muscle fiber is going to be a huge consumer of energy because glucose can be broken apart in the presence of oxygen to give off lots of ATP energy. Other modified structures that we see in skeletal muscle fibers that we'll be going through in more detail later on will include the myofibrils. Myofibrils are the contractile organelles of skeletal muscle tissue. The sarcoplasmic reticulum, and the name sarcoplasmic reticulum should remind you of endoplasmic reticulum, which is a structure you learned in Chapter 3, a series of membrane tunnels found inside the cytoplasm of a cell. And then finally, we will also be looking at the T-tubules, also known as the transverse tubules. Transverse tubules are extensions of the sarcolemma deep into the interior of the cell. So first let's look at the myofibrils, and we're going to spend quite a bit of time looking at myofibrils. Myofibrils are long structures, and they are packed inside an individual skeletal muscle cell and take up a large amount of the cell volume. Myofibrils are densely packed with proteins. So as many of you know, one of the main macromolecules associated with the muscle cell will be protein. Myofibrils are composed out of the sarcomeres. In fact, we're going to find repeating sarcomeres all along the myofibrils. A sarcomere is considered a functional unit of the cell because it is one contractile unit. An individual sarcomere will contract. All the sarcomeres in a myofibril will contract. Therefore, the myofibril will contract because the myofibril makes up most of the muscle cell by volume. Because the myofibrils contract, the muscle cell will contract. The myofibrils and the sarcomeres within the myofibrils will contain many important muscle filaments known as myofilaments. Another structure seen with both skeletal muscle and cardiac muscle is striations, and these striations are due to the arrangement of the sarcomeres within the myofibrils. They're arranged to have perfectly aligned repeating series of dark A bands and light I bands. You can always remember that the A band is the dark band because dark has A in it, and the light band is the I band because light has I in it. So here we can see a small section of a skeletal muscle fiber. On the outside, you can see the sarcolemma. So the sarcolemma is the muscle cell membrane covering the outside of the cell. You can see many, in this view, you can see 3 nuclei pushed up towards the edge. You can see that the interior of the muscle cell is mainly filled with the myofibrils. So they look like long rod-like structures packed inside the muscle cell. The myofibrils will have these little individual units called the sarcomeres. So in this picture, each individual sarcomere is from one blue area, one little blue line, to the next little blue line of that myofibril. And because all of these myofibrils and all of the sarcomeres inside the myofibrils are perfectly aligned, we end up with this alternating pattern of light and dark that you would see looking at the cell underneath a microscope. So the dark area is called the A band and the light area is called the I band. All right, so I'm going to be going through a series of definitions. These are all structures that are seen in the myofibril, specifically in the sarcomere of the myofibril. So we're going to go quickly through this list of definitions, and then we're going to see some pictures of these. And as you look at the picture, you can see where the different structures are. Again, I recommend that you find a blank diagram and have the ability to fill it out and define what the structures are and where they're located. All right, so first we're actually going to start down at the bottom with the sarcomere. As I said, this is the functional unit of the myofibril, the functional unit of the cell itself. It is the contractile unit of the myofibril, but if you're just looking at it in terms of structure, it's the region between 2 successive Z disks. So Z disks are sheets of proteins that anchor together thin and thick filaments. All right, so they anchor the thin filaments in place and they connect one myofibril to another. And the sarcomere is the region between one Z disc and the next Z disc in series. The thick filaments and thin filaments are the main contractile portions of the sarcomere. Thick filaments are physically rather thick. So if you shone a light through a thick series of thick filaments, it would block the light. So if it blocks the light, it's going to appear dark. So the thick filament is the main structure that makes up the A band, the dark band. Thin filament is physically, structurally thinner than the thick filament, and when you shine a light through thin filament area, the light isn't blocked as much, so it will appear to be brighter when you're looking at it through a microscope. The thin filament is going to be found in association with the I band, but some of it will overlap with the thick filament, so it will run partway into the A band. Okay. So the thick filament will actually be found in the very center of a sarcomere. On the edges of the area where you have the thick filament, you have overlap between thick filament and thin filament, but in the very center, you have the H zone, which is a lighter region right in the center of the A band, where the thick filament and the thin filament don't overlap with each other. This H zone also has a line of proteins called myomesin that hook all of the thick filaments together and hold them into place. So let's physically look at a sarcomere, and many of these definitions will make a little more sense once we're looking at this picture of a sarcomere. So a sarcomere will run from one Z disc to the next Z disc. So you can see that the protein that's found at the Z disc, the way it's shaped, it's sort of a little zigzag pattern, hence the name Z disc. The thick filament in this arrangement is the orange-looking thick structure with the little bumpy heads coming off of it. The thin filament is the blue structure that looks much thinner than the thick filament. So if we look at this, we can see that a sarcomere runs from one Z disc to the next Z disc. Two really important proteins found in the sarcomere will be the thick filament and the thin filament. These two proteins will overlap with each other. Everywhere where you see just the thick filament, that's going to be the A band. So that entire area where you see the orange area, so this entire area, let me pick up a drawing tool here. This entire, whoops, sorry guys, I'm trying to draw with some things that don't want me to draw today. All right, so anyway, that entire area where you see the orange, that's going to be the A band. Okay, you can, if you look, you see there's an area where the orange looking thick filament overlaps with the blue thin filament. So there is an area of overlap at the edge, but in the very center, there's an area that's not overlapped, and running down the very center of that is the M-line proteins holding all the thin filaments in alignment with each other. So the sarcomere is the functional unit of muscle fibers. Each sarcomere is aligned along the myofibril like sort of little boxcars, so there's one right after each other. Each individual sarcomere contains an A band and half of an I band. So let's go back to our picture of our sarcomere. As I said, where you see the orange thick filament, that's the A band. Where you see the blue, that's an I band, but if you look to the edge, look at one of the Z discs, you can see that there's some thin filament on one side of the Z disc and some thin filament on the other side of the Z disc. So at the edge of the sarcomere, you have one half of the I band. If you look at the other edge of the sarcomere at the other Z disc, you'll see the other half of the I band. So the sarcomere is mainly made out of the thick and thin myofilaments, and they are what have the contractile proteins. So again, if we look at an entire myofibril, we can once again see this banding pattern, and you can see an individual sarcomere. So find where it says Z disc and the next Z disc. All right, that's one sarcomere. In the central area of that sarcomere, we have the A band. All right. Then if you look at the Z disc, you can see that the Z disc is running right down the center of the I band. All right. In the center of the sarcomere, we have the H zone. That's the area where you only have thick filament, but you don't have thin filament. And down the very center of that H zone, in this diagram, you see three little lines right together. That's called the M line. Those are proteins that hold the thick filament together. So the myofibrils have this orderly arrangement of actin and myosin myofilaments. Actin myofilaments That's the thin filament. All right, the thin filament makes up the I band, and it will overlap a little bit with the thick filament, so it extends partway into the A band. And the actin myofilaments, the thin filaments, will attach to the Z discs. The myosin is the protein associated with the thick filament. So the thick filament is found where you have the A band. And the M-line proteins will help align all of those myosin myofilaments. So the M-line will hold together the thick filaments. So we're going to look a little bit at the structure of the myosin protein. So a thick filament is made up of lots of these myosin proteins bound together. Each individual myosin protein has two components to it. It has a tail area that's fibrous and contains two polypeptide chains that are woven together. And then attached to those polypeptide chains, you have two myosin heads that are globular. They're kind of a spherical protein. And spherical proteins often have binding sites or active sites. These myosin heads are going to reach out and attach to the thin filaments during contraction. But the myosin heads will have two active areas on them. They will have a binding site for actin, and they will have binding sites for ATP. So the binding sites for actin will allow them to form the cross bridge with the thin filament, because the thin filament is mainly made out of actin proteins. The binding sites for ATP are the areas that act as ATP ACE enzymes. That means it breaks apart ATP and you generate a little bit of energy by breaking apart ATP. So here we see, focused in on one section of the sarcomere, let's look over at the thick filament area. The thick filament is in orange. So at the top of the thick filament box, you can see the thick filament with lots of myosin heads hanging off of them. Down just below that, if you follow the arrow down, you can see an individual myosin molecule with the fibrous tail portions woven together. Now look at the heads. There's actually two heads that are bonded together. You can see two different binding areas, an actin binding site and an ATP binding site. So the actin binding site will be where the myosin head attaches to actin, and the ATP binding site will be where the myosin head breaks apart ATP to gain a little bit of energy. Connecting those two areas, connecting the tail to the head region, we have a hinge region. And this is because those heads are going to swivel or pivot, and that swivel or pivot will be a driving force for muscle contraction. Next, let's look a little bit more at the thin filament. The thin filament has a couple of, or three different proteins associated with it. One really important type of protein is known as actin. In fact, there's a whole series of small actin subunits that are found on the fibrous protein actin. They all attach together. Each of those individual actin subunits has an active site where myosin heads can bind. In fact, you'll hear me call these the myosin binding sites. But there are two other proteins found on the thin filament. That is troponin and tropomyosin. Troponin and tropomyosin are regulatory proteins that bind to the actin. They are what will inactivate the muscle fiber. When they cover actin myosin binding sites, that stops contraction. If those binding sites are exposed, contraction can occur. So once again, let's look at the same image of the thick filament and the thin filament. This time, look over on the right-hand side at the box labeled thin filament. If If you look at this box, you can see all the little globular subunits, the little spherical subunits of the actin molecule. If you look at the very bottom where you see each of those individual actin subunits, you see that each of them have an active site where myosin can attach. Then if we look up at the entire thin filament, you see that there are two strings of these actins twisted together. If you look just at those blue actins, you'll see little darker, almost black dots. Those represent the myosin binding sites. Now in this picture, you see tropomyosin, a protein, directly laying over those myosin binding sites. This is going to stop the interaction between the myosin head and the myosin binding site, so contraction cannot occur. But there are some other important proteins associated with the myofibril. I'm going to mention a few of those. The first is an elastic filament known as titin. Titin will hold the thick filaments in place. It prevents them from being torn apart if you stretch the myofibril, but once you've stretched the myofibril, it should spring back into position. So it's going to give the myofibril both the property of extensibility and elasticity. Also, there's a protein known as dystrophin. Now, dystrophin will link the thin filaments, and actually it links the thin filaments at the Z discs up to the proteins of the sarcolemma. So in essence, it essentially links the myofibrils, those organelles inside the muscle cell, to the sarcolemma, the muscle cell membrane. So when the myofibrils begin to contract and they generate force from contraction, they will pull on the sarcolemma, and that causes the entire muscle cell to become a little bit smaller. Now there are some other proteins that are found in the sarcomere, in the myofibril. Nebulin is actually the protein that's found at the Z disc. Myomesin, that's the protein at the M line that holds the thick filaments together. And there are C proteins that will bind many myofibrils together. So most of these, the titin, the nebulin, the myomesin, those are responsible for maintaining alignment of the thick and the thin filaments. I'm going to actually flip back a second because I want you guys to see this protein known as titin. So I'm going to flip back a few slides to one where I have a picture of an individual sarcomere, and I'm going to point out exactly where the protein known as titin is located. So if I flip back, let's go to here. So here's a picture of an individual sarcomere. See the little yellow structure that's twisted up? It actually forms something that looks like a spring. It attaches to a Z disc, and then it also attaches to the thick filaments. So as it goes through the Z disc, it ends up connecting to a thin filament, and then goes all the way across and connects to the thick filament. So that is titin, an elastic filament. And those little circle areas, it literally does form a spring. And just like a spring, you can stretch it out. It is extensible. But then when you let go, it will spring back to its normal length. It is elastic. All right, let's continue on with some of the specialized organelles found in the muscle tissue. First, we have sarcoplasmic reticulum. Sarcoplasmic reticulum is a network of endoplasmic reticulum that will wrap around each individual myofibril and run longitudinally. That means it runs along the myofibril. In this case, this is a special form of smooth endoplasmic reticulum. This smooth endoplasmic reticulum stores the ion calcium. So it will store calcium and keep it physically away from the myofibril. When it stores the calcium and keeps it away from the myofibril, that's what you see when the muscle cell is relaxed. But when you trigger contraction, you're going to release the calcium, and the calcium is going to be the trigger for the T-tubules are another example of a modification that we see in the skeletal muscle fiber. We also see this modification in cardiac muscle, although it's not found in smooth muscle. T-tubules are extensions or continuations of the sarcolemma. So remember, when we say sarcolemma, we're usually talking about the membrane up at the surface of the muscle cell. T-tubules are little extensions that go inside of the cell. Think of it as if you had a balloon. A balloon has a fairly flexible membrane. Now blow the balloon up in your mind, and then take your finger and then just push down into the balloon. That's like a T-tubule, except there are many of them that form networks all throughout the cell itself. So the interior of the T-tubule, the lumen, is continuous with the extracellular space. Now later on, when we talk about some muscle physiology, we'll see the movement of ions from the extracellular fluid into the muscle cell itself. So this also increases the muscle fiber's surface areas, the surface area across which these ions can move, and that material, those ions from the extracellular fluid, can be moved deep into the interior of the myofiber, the muscle cell itself. So here we can see some of these special modifications. In white or maybe kind of gray, you can see the sarcolemma on the surface, and then the T-tubules that go in from the sarcolemma and encircle each individual myofibril. Also surrounding the myofibrils, and the myofibrils are in orange, encircling the myofibrils, you see the sarcoplasmic reticulum. SR stands for sarcoplasmic reticulum. This is the area that will store calcium. Now notice that there is an enlarged area of sarcoplasmic reticulum right next to a T-tubule. This is intentional. The T-tubules are going to produce a stimulus that will cause the sarcoplasmic reticulum to release calcium. So most of the storage, the cistern is a storage area. Most of the storage of calcium in the sarcoplasmic reticulum is in an area right by the T-tubules. And one last thing I want to point out on this side is that there are lots of mitochondria found in the muscle cell. The mitochondria are usually lined up right alongside of the myofibrils. Mitochondria are the powerhouses of the cell. This is where glucose is completely broken apart in the presence of oxygen to produce lots of ATP energy. So that does it for this lecture on the microscopic structure of the muscle cell. The next topic we're going to get to in the next video lecture will be the sliding filament model of contraction as we begin muscle cell physiology.